Prostate cancer. These four have it. Many of us know people who have it, friends, family. It's very common, and in fact, in the UK, about 1 in 8 men in their lifetime will be diagnosed with this particular disease. And in 2020, it became the most commonly diagnosed cancer in England. So it is a serious problem. We know the risk factors for it, broadly speaking. We know that the risk increases with age. The risk increases with ethnicity. If you are of African descent, you are more likely to get prostate cancer during your lifetime. And if you have first-degree or second-degree relatives, then there is a good chance you might have to screen regularly for the disease as well. But what about infection? Are there any other risk factors? Could infection be a risk factor? Because we know that when it comes to cancer, we have examples where bacteria and viruses have caused different types of cancer, including gastric and cervical. So the question then becomes, could bacteria also be involved in prostate cancer? That was the basis of this paper from the Brewer group, of which I'm a part of. So this was just before I started my PhD 4 years ago. And essentially, the take-home message from this is that the presence of certain bacteria in urine is associated with an increased risk of developing aggressive prostate cancer. Which bacteria? That leads us to the topic for today, the anaerobic bacterial biomarker set. It's a group of 6 bacteria. Not expecting you to remember the names, but just remember that there are 6. Phenolaria, Escherichia, Peptinophilus, Anaerococcus bophrimonas, and Fusobacterium. And in the paper from which these figures have been taken, it was shown that the presence of these organisms within the tumor microenvironment led to an increased risk of developing aggressive disease early on. But that's not an exhaustive list. And my question was, are there more bacteria that belong to this particular group, which we just haven't identified? And in order to do this, I'm looking at a large data set from which I'm going to be trying to figure out if there are more such bacteria that need to be looked at. And that is the pan-prostate cancer group dataset, which is a dataset of more than 2,000 tumors and matching blood samples. And it has whole genome sequencing data from around the world, from 6 countries to be precise. And essentially what I'm looking at is that last line which says human depleted FASTQ files of more than 900 primary tumor samples, which essentially means that the tumor samples have been depleted for human reads, so whatever is left is microbial in origin. And in order to evaluate whether or not there are such bacteria, one of the important tools that I'll be sort of touching on more often than the others is something called HUMAN3, which is the HMP Unified Metabolic Analysis Network. And essentially, the aim of this tool is to profile metabolic pathways and gene families from sequencing data. It gives you 3 output files, at least the version that I'm using. They've since updated it to remove path coverage. But what is important for our work is the gene families. That is the characteristic that we will be using. My approach to this problem was threefold. So the first one involved looking at the phenotypic characteristics from sequencing reads. And in order to do that, I use something called Trata, which is another software. And the list of organisms that I've chosen to look at for their phenotypic characteristics come from the paper that was published, as well as from another part of my PhD, which is where I got the organism Winkia newi from. I then get the list of organisms that are similar to ABS by their phenotypic traits. I then do a similar sort of process for gene families as well. And finally, I use BACTA for genome annotation of the same set of candidates that I used for Trata work, and get a list of organisms that are similar by genome annotation as well. Combine the 3, you get a short list of how many ever. And essentially what I'm looking at after that is to see if they have any impact on survival. And when I say survival, I just mean biochemical relapse, which is what BCR is. So I've looked at it in 855 samples, and I'm hoping that at the end of all of this, I get a final list of candidates. So with Trata work, what it does, just as an example, is it highlights the differences between your ABS bacteria and your non-ABS bacteria or your candidate bacteria. And The bar plot on the top right essentially tells you the phenotypic traits that are the most important in terms of differentiating these two groups. So you have things like gram negativity, which is a characteristic of ABS, and other traits as well. When you plot them onto a heat map for all of the candidates, which are on the x-axis, and then you have the traits on the y-axis, this is kind of what you get, and it's a simple presence absence. Does this particular bacteria have that trait or not? And all of it's based on sequencing reads. We then plot this onto an NMDS plot with Jacquard distances. And what you can see there are the ABS organisms that are highlighted in red, and everything else is candidates. And in order to filter this list out and get a short list, we have a distance threshold of 0.4. Similar sort of thing with gene families, and essentially what this shows you is a heat map of gene family presence and absence, and basically any genus that has an average similarity of more than 0.5 to ABS was taken. Again, gene families was the output that was taken from human. And finally, genome annotation. Genome annotation was done with Bacta, and essentially gives you a similar plot to what we've seen before with Treta work, and because it's the same set of candidates. And essentially what you see is how closely related the candidates are to your ABS bacteria. Again, the same distance threshold was set so that any genus closer than 0.4 to ABS was taken forward. So once we've got all the shortlists, we then get 9 candidate genera. Now, at this point, I do have to say I can't actually name any of the candidate genera because of some IP issues that are well above my pay grade. So I can't actually name any of them. So I just had to come up with very creative names such as genus 1. Yeah, but I have compared. So I did still move forward, and with my data, I did look at if they are associated with a biochemical relapse of prostate cancer. And as you can see, genuses 1 to 5, if they're present, which are indicated by the red line, tend to sort of be present in patients who have worse outcomes than patients who don't. So yeah, essentially I've labeled them ABS 2.0. Which are 5 genera that are extremely similar to ABS based on their phenotypic traits, based on gene families, and based on genome annotation, and have an association with the difference in relapse-free survival. The next question then becomes, are they present in the same samples as the original ABS, which is what I looked at. And what I found was that they tend to co-occur a lot with Fusobacterium and Porphyromonas, although there is some amount of co-occurrence in general with all of the ABS genera. And yeah, the last thing that I wanted to look at was whether when you group all of these together, the original ABS and the new ABS, or ABS 2.0, does it affect BCR? So first I looked at just the original ABS, and what I found was that over the course of time, patients with ABS tend to have worse prognosis, which just confirms what was said before. But when you then group ABS and ABS 2.0 together, so now there's 6 from the original, 5 from the new one. So when you group 11 of them together, what we find is that when you stratify them according to increasing bacterial load, you then get this survival curve where over time, the more number of these 11 bacteria are present in a patient, the worse the outcome, which is kind of what I expected before I did this, and that's what was found. So to conclude, there's very strong evidence for 5 new genera to make up ABS 2.0 because they share common phenotypic characteristics, share a high degree of similarity in their gene family profile, and their genome annotation indicates similarity at genome level as well. They seem to co-occur a lot with Fusobacterium and Porphyromonas, which might be something interesting to look at. One thing which I haven't shown here is these effects are seen with biochemical relapse, but when we look at metastasis as the endpoint, these effects are not replicated. My guess, and this is purely an educated guess, is that there is some role that treatment plays into it, and it could be due to that. The other thing I want to stress is just because these bacteria are present in these patients doesn't mean they're the reason they're progressing a lot faster. So I do want to stress that correlation is not causation. And finally, I think the next step is, apart from all of this, is to characterize them at species level and find any potential mechanisms where that might explain all of these findings. I'd like to thank my supervisory team, my funders, and thank you for listening.